Welcome back to G Fun Facts Online. Today we are diving deep into a technology that sounds like it was ripped straight from the pages of a cyberpunk novel, but is actually happening in laboratories right now. We are talking about optically printed bioelectrodes. If you have ever imagined a future where humans and machines integrate seamlessly, where we move beyond clunky robotic limbs or rigid pacemaker boxes to something that feels natural, organic, and invisible, this is the science that is going to get us there. We are going to skip the fluff and get straight into the engineering, the chemistry, and the incredible physics behind merging light and biology. To understand why this is such a massive breakthrough, we first need to dissect the problem with the medical electronics we use today. Right now, if you need a neural interface or a heart monitor implanted, surgeons are essentially putting a rock inside a bowl of jello. The human body is soft, curvilinear, and dynamic. Your skin stretches, your heart beats, your lungs expand. In stark contrast, traditional electronics are rigid, planar, and static. Silicon wafers, the foundation of modern computing, have a stiffness or Young's modulus of about 130 to 185 gigapascals. Brain tissue, however, is incredibly soft, sitting at around 1 to 3 kilopascals. That is a mechanical mismatch of 6 orders of magnitude. It is roughly the difference between a block of granite and soft gelatin. When you implant a rigid silicon or metal electrode into the soft tissue of the brain, every tiny movement of the head causes microfriction between the device and the neurons. Over time, this mechanical irritation triggers the body's foreign body response. The immune system attacks the intruder, and glial cells wrap the electrode in a scar, insulating it from the very neurons it is trying to listen to. This glial scarring is the primary reason why neural implants like the famous Utah array often fail after a few months or years. But the mismatch isn't just mechanical, it is also chemical. Biological systems communicate using ions like calcium, potassium, and sodium. Electronic devices, however, communicate using electrons. This creates a translation gap. To bridge it, traditional metal electrodes made of gold or platinum rely on capacitive coupling or reactions at the surface. The problem is that these interfaces often have high impedance, which means high resistance to the alternating currents of the body. This results in noisy signals. To get a clear reading, you historically needed large electrodes, which limits the resolution of the data you can collect. On top of all that, there is a fabrication bottleneck. Making computer chips requires photolithography, a process involving harsh toxic solvents that dissolve organic matter. High temperatures, often exceeding 100 degrees Celsius, which would cook biological cells, and expensive vacuum chambers. You simply cannot put a living heart or a delicate plant leaf inside a vacuum chamber and spin coat it with photoresist. This limits us to fabricating devices on plastic sheets and then trying to paste them onto the body, which is far from ideal. This is where optically printed bioelectrodes come in to solve the mechanical, chemical, and fabrication problems in one stroke. The core innovation here is something called visible light photoinduced polymerization, or VLIP. The star of this show is a material called PEDOT. Unlike copper or gold, PEDOT is an organic conductive polymer. It conducts electricity, but it is soft, flexible, and processes like a plastic. What makes PEDOT truly magical is that it is a mixed conductor. It conducts both electrons, like a metal, and ions, like a salt solution. This makes it the perfect translator between the body and the machine, lowering impedance by orders of magnitude compared to metals. So how do we print this stuff directly onto biology? In the past, synthesizing PEDOT required harsh chemical oxidants like iron chloride. It was a messy, toxic process, but researchers, particularly those at institutions like Link and number 246 Sping University, developed a photosensitive approach. They created an ink solution containing water-soluble monomers, which are the building blocks of the plastic, mixed with a photocatalyst. The process is elegant in its simplicity. You apply this ink to the target surface, whether that is glass, plastic, or even living tissue. Then, you hit it with a beam of visible light, like a blue or green laser. When the light hits the ink, it excites the photocatalyst. The catalyst steals an electron from the monomer, creating a radical. These radicals then attack other monomers, chaining them together into a long, conductive polymer chain. The result is a solid, conductive electrode pattern that forms only where the light touched. The remaining unreacted liquid is simply washed away with saline. The advantages here are massive. First, the conditions are mild. There is no heat, no vacuum, and no toxic solvents. It happens at room temperature in an aqueous environment, which is friendly to cells. Second, because it uses light, the resolution is limited only by your optics. Techniques like two-photon polymerization can achieve features as small as 100 nanometers, which is smaller than a single bacterium. Third, it is conformal. You can project a pattern onto a curved surface, like a nerve bundle, and the electrode will form perfectly to that shape, ensuring optimal contact. Let's dig a bit deeper into the physics of why these electrodes are better at listening to the body. It comes down to the electrochemical interface. A traditional metal electrode interacts with the body only at its 2D surface. But a PEDOT electrode acts like a sponge. It is porous and permeable to ions. When a neural signal, which is an ionic wave, washes over it, the ions soak into the volume of the polymer. This concept is called volumetric capacitance. It means that a tiny speck of PEDOT can store and transfer as much charge as a much larger piece of gold. This massive effective surface area drastically lowers impedance. In EEG recordings of brainwaves, optically printed PEDOT electrodes have demonstrated significantly higher signal-to-noise ratios than standard metal electrodes, allowing them to detect weaker, more subtle brain activity. Furthermore, they lower the contact barrier. Dry metal electrodes often face high resistance at the skin interface, sometimes hundreds of kiloohms. Optically printed polymers are soft and chemically similar to biological tissue, forming a much more intimate wet contact. This drops impedance, often below 10 kiloohms, without the need for those messy conductive gels that dry out over time. Now, let's talk about the different ways we can actually print these things. There are a few main techniques. First, there is projection stereolithography, or DLP. This is similar to resin 3D printers you might have seen. It uses a digital projector to flash an entire layer of the electrode pattern at once. It is extremely fast and can pattern large areas, like a whole electronic skin patch, in seconds, though the resolution is generally limited to the pixel size of the projector, usually around 10 to 50 microns. Then there is laser-induced forward transfer, or lift. This is a digital stamping technique. A transparent donor plate is coated with the conductive material. A laser pulse hits the back of the plate, creating a tiny bubble that propels a droplet of the material onto the receiving tissue. This is great because you can print sensitive biological materials, even living cells, alongside the electrodes. Finally, there is two-photon polymerization, the sniper rifle of optical printing. It uses a femtosecond laser focused to a tiny point. Polymerization happens only at the exact focal point, where two photons hit a molecule simultaneously. This gives you incredible 3D freedom, allowing you to print hollow needles, scaffolds, or complex lattice structures inside a hydrogel or tissue sample. So what can we actually do with this technology? The most immediate impact is in medicine. One of the most groundbreaking demonstrations involved researchers applying a monomer gel to the exposed brain of a mouse. They used a laser to draw electrodes directly onto the cortex. The electrodes formed an intimate, seamless contact with the brain tissue, recording neural spikes with clarity that rivaled penetrating needles, but without piercing the brain. This could lead to shrink-wrapped brain interfaces for treating epilepsy or controlling prosthetic limbs that grow with the brain rather than constricting it. Then there is wound care. Imagine a burn victim. Instead of painful dressing changes, a doctor sprays a monomer solution onto the wound and uses a handheld blue light to print a sensor mesh right on the skin. This mesh could monitor temperature, pH, and infection markers in real time. If an infection is detected, the electrodes could even be triggered to release antibiotics stored within the polymer matrix, or use electrical stimulation to accelerate wound healing. We are also looking at cardiac wraps. The heart is the most mechanically dynamic organ we have. Rigid pacemaker leads can fracture or damage heart tissue. But optically printed conductive elastomers can be printed directly onto the outer layer of the heart. These cardiac socks can map the heart's electrical rhythm in high resolution to diagnose arrhythmia and deliver pacing shocks exactly where needed, using lower voltages that are less painful and damaging. But this cyborg concept isn't limited to humans. The field of plant bioelectronics is exploding. Researchers have created what they call the cyborg rose. They placed a rose stem into a solution of EDOT monomers. The plant's natural vascular system, the xylem, sucked up the solution. Inside the stem, the natural chemistry of the plant polymerized the monomer. The result was a rose with conductive wires running through its stem, formed by its own biology. These e-plants can act as sensors, monitoring environmental pollutants or drought stress from the inside out. In precision agriculture, optically printed sensors on leaves could monitor transpiration and photosynthesis in real time. Instead of watering a whole field based on a schedule, a farmer could water individual plants based on the electronic distress signal sent by their leaves. Now, if we want to build a real biocomputer, we need more than just wires, we need transistors. The organic electrochemical transistor, or OECT, is the biological equivalent of the silicon transistor. It uses a gate voltage to control the flow of ions and electrons in a channel. These are incredible amplifiers, capable of taking a tiny biological signal and boosting it thousands of times right at the source. Recent advances have moved from printing just wires to printing full OECTs. By using light to pattern the source, drain, and gate, researchers are creating logic gates like AND, OR, and NOT on flexible films. The vision is to create a biocomputer printed on a sticker that processes health data locally on the skin, without needing to send raw data to a smartphone, saving massive amounts of power. Of course, we have to be realistic. There are still significant challenges to overcome. The first is stability. Organic polymers are sensitive. UV light, oxygen, and moisture can degrade PEDOT over time, causing it to lose conductivity. While self-doped variants and new cross-linking strategies have extended lifetimes to months, medical implants need to last decades. Researchers are working on encapsulation strategies and hybrid materials that mix PEDOT with carbon nanotubes or graphene to boost durability without sacrificing softness. Then there is the washout problem. When printing in a living body, you must ensure that unreacted monomers, the ink that didn't turn into plastic, are flushed away completely. While PEDOT is relatively safe, high concentrations of any foreign chemical can be problematic. Developing 100% conversion chemistries or bioresorbable monomers is a hot area of research. And finally, there is the interconnect problem. You printed an electrode on a brain, which is great, but how do you plug it in? Connecting a soft, micron-thin polymer film to a hard copper wire in a battery pack is a mechanical nightmare. This is often where flexible electronics fail. Solutions involving wireless power and data transfer, or gradient connectors that transition slowly from soft to hard materials, are currently being developed. Despite these hurdles, optically printed bioelectrodes represent a philosophical shift in engineering. We are moving away from imposing machinery onto biology, and toward growing technology within it. As we refine the chemistry of these light-activated polymers, the applications will become even more exotic. From regenerative bioelectronics, where printed scaffolds guide stem cells to regrow severed spinal cords, to edible electronics that monitor gut health and digest safely. The marriage of light and biology has produced a soft, conductive, intelligent material that promises to heal, monitor, and connect us in ways we are only just beginning to imagine. Thanks for listening to this deep dive on Fun Facts Online.